Hi biologists, let's start by looking at the learning objectives for this lesson. At the end of this lesson and following the biology syllabus, you should be able to give a detailed account with the aid of diagrams of the stages of mitosis. What does this actually mean? What are we trying to learn? Well, it's straightforward enough. You have to be able to describe in detail, with the help of diagrams, the process of mitosis. Let's look at a detailed study of mitosis. You might remember the cell cycle from a previous video. The cell cycle, or the life cycle of a cell, can be divided into stages. Interphase is the stage in the cell cycle during which cells are not dividing. Mitosis is the division of the nucleus to produce two genetically identical daughter cells. Because this is a detailed study, you must also understand that mitosis is usually followed by cytokinesis or cell division. This is where the cell or the cytoplasm divides to form new cells. Now, strictly speaking, the objective of this video is to have a detailed understanding of the process of mitosis or the process of nuclear division. An understanding of the cell cycle stages helps the understanding of mitosis. So, a detailed study of mitosis involves watching the following videos in the sequential order given. Mitosis number 1, which describes the process of interphase. Mitosis number 2, which involves a detailed study of mitosis, and mitosis number 3, which describes the process of cytokinesis. Let's look at mitosis or nuclear division. Let's begin by revising the definition of mitosis. Mitosis is nuclear division, where the nucleus divides to form two new nuclei, each containing the same number of chromosomes with identical genes. Make sure you learn the definition of mitosis. Mitosis means that two new genetically identical cells are produced, two new genetically identical nuclei, if you like, are made with the same number of chromosomes as the original nucleus or the original parent cell. These two new genetically identical cells are clones of each other. Now, the result of mitosis is, as we've just said, the formation of two new identical daughter nuclei. The word daughter here does not refer to female. It simply means offspring nuclei or two new cells. The original cell, or the mother cell, and the two daughter cells have the same number of chromosomes, the same number of genes on those chromosomes, the same type of genes as each other. So as we've just said, the original cell and the two new cells formed are clones. Now, an interesting point for those of you able for a bit more is that in mitosis, haploid cells produce haploid cells. So in other words, if you start with a cell with a single set of chromosomes, you will end up with two new cells, each of which also have a single set of chromosomes. If you start with a cell where the chromosomes are in pairs, or a diploid cell, then two new diploid cells will be produced, each of which have two sets of chromosomes, or chromosomes in pairs. Now, mitosis occurs in somatic cells. These are cells not involved in reproduction. In animals, mitosis takes place throughout the body. If you've studied the skin, you might have come across the Malpighian layer in the skin. This is a region where new skin cells are formed through the process of mitosis. We can remember this because the M&Ms, M for mitosis, M for Malpighian layer. In plants, mitosis occurs in special or specific growing areas, the tips of roots and the tips of shoots. In other words, mitosis occurs in meristematic regions. Again, we see the M&Ms, M for meristematic and M for mitosis. We must realize that mitosis is a continuous process, rather like a film, with no stop for the raffle halfway through the show. It's a continuous process. However, it is divided into 4 stages for convenience or for study. It's very hard to have a clear division between each stage, so that's why, depending on what book you read, there might be slight differences in the account of mitosis. There are 4 stages of mitosis, as we have just said. The 4 stages are prophase, metaphase, anaphase, and telophase. We will work our way down through each of these stages in turn. Don't forget, interphase is not a part of mitosis. It is part of the cell cycle, and it precedes, or it's a prologue, or it goes ahead of mitosis. And a lot of activities go on in interphase that are important for mitosis, but as such, it is not a part of the process of nuclear division. Now, when we look at prophase, we realize that the chromatin threads, IN in chromatin, IN in invisible. You can't see these threads at the moment. You can't see these chromosomes. So the first thing that happens in prophase is that these threads become visible. When the threads become visible, you realize that the chromosomes are actually double-stranded. You have 2 chromosomes. They have photocopied, or if you prefer, technically, the DNA has replicated, creating 2 identical DNA strands. Now, this process happened in interphase, strictly speaking, but it's only in prophase that you can see the results of the DNA replication. This helps us remember prophase. P for prophase, P for photocopying. You can see the photocopying, you can see the double-stranded chromosomes. They are joined at the centromere. The centrioles move to opposite sides of the cell. Again, we can remember this because opposite sides of the cell quite often are referred to opposite poles, like the North and the South Pole being opposite to each other. And we have 2 P's in the word opposite, and we have 2 P's in the word prophase. So far, we can see the DNA replication. We can see that the chromosomes are double-stranded, that they have photocopied, and we can see in prophase that the centrioles move to the opposite sides of the cell. The nuclear membrane also starts to break down. In metaphase, the chromosomes will move and line up across the middle of the cell. They line up along the equator. The equator is a science word for a straight line. Hence you have the equator across the centre of the planet. In metaphase, the fibres which are produced by the centrioles will attach to the centromeres. And finally, the nuclear membrane is fully broken down. Just bear in mind that I like to draw the diagram like this, with the straight line going from top to bottom, showing that the chromosomes are lined up across the middle of the cell in that direction. But you must realize if I flip the picture over, then it can equally well be drawn in that manner, where the chromosomes are lining up more left to right along a horizontal line, more like an equator. Keep this in mind when you're trying to identify pictures of metaphase. The next phase is anaphase. Think of the Latin word annulment, an, to separate, to annul. So in this phase, the chromosomes are going to separate. How does this happen? Well, the spindle fibres will contract. I like to imagine them like a tug-of-war rope, and they will pull. So how this works is that they actually shorten or contract. When they shorten or contract, then the sister chromosomes will be pulled to the opposite sides of the cell. Those of you who are sharp-eyed should now be realizing that we have a set of 4 chromosomes now here on the left and a set of 4 chromosomes here on the right, because it's vital to appreciate the fact that at the end of this process we are going to end up with 2 new nuclei identical to each other and to the parent cell. So in a nutshell, in anaphase, the chromosomes are pulled apart. A and P for apart, A and P in anaphase. Reminding you that identical genes or chromosomes will be pulled to each end of the cell. And again, I like to draw the diagram this particular way, but if I flip the picture over, then I can have a group of chromosomes pulled to the top of the cell and a group of chromosomes pulled to the bottom of the cell, instead of being pulled from side to side, left to right, the way I like to draw my diagram. The final phase of mitosis is telophase. Now in telophase, the spindle fibres break down. The chromosomes become chromatin. We can see these chromosomes at the moment, but they will start to elongate and start to become invisible. Don't forget, IN in chromatin and IN in invisible. The nuclear membrane will reform around each group of chromosomes, and as a result you will end up with 2 new nuclei. 1 nucleus here on the left and 1 nucleus here on the right. And you might see 1, 2, 3, 4, the exact same number of chromosomes present in the second cell, 1, 2, 3, 4, hence creating 2 identical nuclei, T for 2 and T for telophase. So as we have said, there are 4 stages in mitosis: prophase, metaphase, anaphase and telophase, not forgetting that interphase as such is not part of mitosis. Now your problem is, for the exam, that you have to be able to associate each phase with its corresponding picture, and you have to be able to write an account of what's happening in each phase. So, as a heads up or a memory tip, we remember what happens in prophase because we have P for photocopying and we can see that the DNA has replicated or duplicated. We can see the photocopies. We also remember that the centrioles move to opposite sides of the cell. So they move, in my particular diagram, to the left and to the right. Hence we have the centrioles are moving to the opposite poles. 2 P's in opposite, 2 P's for poles, 2 P's in prophase. In metaphase, the chromosomes line up in a straight line across the middle of the cell, otherwise known as the equator. So you have M for middle of the cell, E for equator, and E for that straight line. The next phase being anaphase, we have A for anaphase, A and P for pulling apart, so the chromosomes separate, moving left and right in my diagram. And finally, telophase is where you end with 2 new daughter nuclei, each genetically identical to each other and to the parent cell. So this is easy enough to remember. T for 2 and T for 2 nuclei and T for telophase. And there you have it. Practice in a jotter. Write out the account. Draw out the diagrams. Do it on the double. Now that we have reached the end of our lesson, have we achieved our objective? Can you give a detailed account with the aid of diagrams of the stages of mitosis?